for reaching on our last two crash courses. Class, for Crash Course 9, we are going to talk about your very favorite process in your biochemistry. Diba? Your central dogma of molecular biology. Actually, class, this topic is much more related in your biology class. So let's check the content for our discussion here. Of course, similar with our other crash courses, we will have an overview discussion. In particular, we will discuss your DNA organization. All right. Secondly, of course, that will start our different processes under your central dogma. In particular, replication, transcription, and translation. Class, don't forget, for transcription and translation, we have added your different post-process modification. Okay? Don't worry, we will discuss that in detail in our discussions here. And for our last topic, we have your mutations. Ayan. So class, before I forgot pala, I divided our discussion for central dogma into two different videos. So for part 1, we are going to discuss the overview and your DNA replication. For part 2, we will discuss your transcription, translation, and mutation. So class, without further ado, let's start our discussions here with our quick recap. Ayan. Remember, diba? We already discussed biomolecules, their structure, function, and properties. Check. And then of course, your cellular respiration, including your glycolytic pathway, citric acid cycle, and electron transport chain. Check. And then of course, we also discussed your carbs metabolism. Done na tayo. And for the past two videos, we discussed your lipid metabolism and your amino acid metabolism. Nagka-amnesia na naman ba? Don't worry class, we will just brush up your knowledge on these topics on our succeeding slides. So class, dapat at this point, little by little, may progress tayo on your review here on biochem. So I know that the information, diba, the nature of your biochem is really volatile. That's why you really need to take or exert an effort in order to familiarize all of the essential concepts here. Right? Ayan. So class, of course, again, it's important that you allot ample time in order to review, revisit our discussion, notes, the slides that we are sharing to you class. Ayan. So again, for our quick recap, ayan, done na tayo sa lipids again and your amino acid metabolism. For our quick recap, again, we have your beta oxidation, or our general term is your fatty acid metabolism, diba? So here, our process includes the degradation, or particularly the oxidation of fatty acids in order to produce acetyl-CoA. So it will be fed in your TCA cycle. That's why it is a catabolic pathway in order to generate energy. Or recall, majority ng catabolic pathway again occurs inside the mitochondria. And here, class, I have emphasized that the committed step for our catabolic pathway in fatty acids is your transferase or your carnitine-mediated entry via your acyltransferase enzyme. And here are the energy products. So diba we calculated the total ATP or the net ATP yield of these three products when they were fed in your TCA cycle and in your electron transport chain. Class, please remember the formulas, all right? Second, ayan, so essentially this is just the reverse of your oxidation process. So here, check. From acetyl-CoA, we will generate or produce your fatty acids. So building up siya class, that's why anabolic po siya. All right? O sir, ano nga po yung difference between these two? Again, in order to avoid a futile cycle of your oxidation and your biosynthesis, we use different sets of enzymes, coenzymes or cofactors, and other electron carrier, diba? So for the location, of course, anabolic, majority of them occurs in your cytosol. And for our key enzyme, naaalala pa si ACC, so that's the enzyme responsible in catalyzing the conversion of your acetyl-CoA to its activated form, malonyl-CoA. Diba? Si acetyl-CoA, carboxylase, ang committed enzyme or our regulated step in our biosynthesis pathway. And of course, our natural product is your 16-carbon or your even-numbered unsaturated fatty acid. Okay. For our next pathway, ketogenesis, o sir, di ba alternative energy source yan? Yes po. So si ketogenesis, it acts up in order to store your energy first for utilization on the future. Okay? Si ketone bodies will be synthesized here. That's why it is a catabolic process because this is an alternative source of your energy. And of course, if it's catabolic, ayan, it's in your mitochondria. And recall, the key enzyme is the one converting our intermediate to your HMG-CoA. And recall, our ketone bodies here, the two main ketone bodies again, acetoacetate and hydroxybutyrate will be processed or will be metabolized by our extrahepatic tissues when energy is needed. Okay? O sir, how about acetone? Class, diba? Did you know it is exhaled? Ayan. And lastly, we briefly discussed cholesterol biosynthesis. Again, similar po ang precursor molecule ng biosynthesis, ketogenesis, and cholesterol biosynthesis. Diba? So here, we use acetyl-CoA. And of course, if we do not need that energy immediately, we will alternatively funnel your acetyl-CoA to your cholesterol biosynthesis to form our steroidal compound cholesterol. That's why anabolic, check, it occurs in your cytosol as well as in your smooth ER for post-processing. So we are including here, or we are adding in your smooth ER, your methyl junctions ng cholesterol. And class, recall, your HMG-CoA reductase catalyze the conversion of HMG-CoA to your mevalonate. Diba? So class, I emphasized mevalonate because again, that's a board exam question. Let's move forward with our amino acid metabolism. First, degradation. So class, again, degradation, metabolic pathway are just similar terms. Okay? So from degradation, ayan, we will able to generate your gluconeogenic or your TCA cycle intermediates mainly for energy source. But always remember, priority ng biological system, si carbs, and then si lipids, then last priority, si amino acids as energy source. Alright, for metabolic pathway or type rather, it is catabolic and it occurs both in your cytosol and mitochondria. Diba? For the key enzymes, we have two types of aminotransferase, your ALT and your AST. Diba? I required you to memorize the meaning of SGPT and SGOT. Okay, so class, another key enzyme here is your glutamate dehydrogenase, responsible in majority of our transamination reaction. Ayan, so class, remember the products. First one is your source ng carbon skeletons, that is your alpha keto acid. And of course, our ammonia is our carrier ng nitrogen for expression in the urea cycle. O, diba? Ayan, for urea cycle, of course, detoxification of your ammonia via urea excretions. Of course, this pathway is amphibolic. It creates or it produces TCA cycle intermediate, and it is also catabolic in terms of its disposal mechanism. O class, unahan ko na kayo. Again, all locations here in our amino acid metabolism occur in both cytosol and mitochondria. Okay, so we summarize the cellular locations of all metabolic pathways on our last video as well as their metabolic types. So, diba? For our key enzymes, so moving forward, we have the enzyme catalyzing the production of your carbamoyl phosphate. So that's your CPS1 or your carbamoyl phosphate synthetase 1. And of course, the product is for expression, urea. Ayan. And lastly, the amino acid biosynthesis can either be through recycling or through de novo biosynthesis. O, diba naaalala? Okay, for our metabolic type, of course, that's building up of our amino acids. So obviously, class, that's an anabolic pathway, again, occurring in both of your cytosol and mitochondria, particularly sa liver cells natin. Ayan, for key enzymes, of course, glutamine synthetase, check, and your numerous transaminase or aminotransferase enzymes. And products are your, again, class, non-essential amino acids. Recall, your essential amino acids cannot be synthesized in our biological systems or by our cells. Okay? Class, clear na ba on your lipid and amino acid metabolism? Ayan, so for this video, again, we will start the discussion na on your central dogma with this introductory video on the processes occurring in our central dogma. Let's watch this short video.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones.
Class, that's actually our DNA organization in your prokaryotic cells particularly. Don't worry, we will discuss this in detail later on our discussion on DNA organization. Again, very different ang organization in your prokaryotic and eukaryotic cells. All right.
Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA.
Class, quick quest tayo. Again, this process is what we call so from your DNA, we will transcribe the gene in order to form your mRNA. Of course, that's your transcription. But remember, hindi exclusive transcription with your mRNA. So we can actually synthesize a lot of types of RNA in our transcription process. Ayan.
This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA.
Class, recall from your undergrad biochem, what is this process called? So after replication, you have your transcription, and finally, to convert your mRNA, your transcript, to your proteins, we call this process your translation. Diba? So site of our translation or protein synthesis is in your ribosome in the cytosol. All right.
The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Alright, so class, that's it for our introductory video here in our central dogma. Okay, you already seen our discussions for today. First, of course, your replication, then transcription, and then translation. So first, we should discuss the structure of our DNA packing in our nucleus. Alright, so remember from our nucleic acid discussion, your DNA structure is stable because of first, the base pairing, diba? And of course, ayan, we know that aside from base pairing of your nitrogenous bases, in the sugar phosphate backbone, the stabilizing interaction is your phosphodiester bond. Alright, and aside from that, we also have your base stacking interaction. That's between base pairs in order to efficiently compact our DNA in our chromosomes. And aside from that, of course, we also have binding sites in our DNA in your major groove and minor groove. So majority ng ating transcription and your replication factors, of course, they bind in your minor groove. Alright, so class, I hope that you still remember our discussion on DNA structure because, again, in your chromosomes, our DNA is the building block of our genetic structure. Alright, so more particularly, ayan, DNA is packaged differently in your eukaryotic and prokaryotic cells. So let's check. Ayan, for our prokaryotes, sir, walang nucleus si prokaryotes. Yes, that's why it's only in your nucleoid because, again, wala siyang true nucleus. Again, the location of the DNA of your prokaryotes is in your nucleoid. For our structure, ayan, single circular chromosomes lang ang structure niya. And of course, in terms of its packaging, purely DNA lang siya, class. Okay? So for eukaryotes, ayan, in order to protect the structural integrity of your DNA, of course, it should be inside the nucleus with linear chromosomes. Sir, akala ko X, class, of course, your chromosomes has different shapes or your structures rather. Okay? So yung X, yung distinct like X structure niya is in your cell division or before cell division. Alright? And for packaging, remember, here in our DNA organization, your DNA actually wraps around histone proteins. Class, later we have a dedicated slide for your histone proteins because this appeared already in the board exam. Okay, so let's check the structure of your prokaryotic DNA. Ayan, single and circular chromosome lang siya found in your cytosol or your nucleoid. Okay, so again, class, pure DNA, no association with other biomolecules. For our eukaryotic cells, of course, as we have said, linear chromosomes. Ayan, and of course, it is wrapped around your histone protein. So of course, class, ang certain region ng chromosome, we call it your chromatin. So very important in structure ng ating chromosome in order to regulate the access of your major and minor groove that are very vital in your replication, transcription, and repair mechanisms of our cells. Let's dissect our structure of your DNA in prokaryotic cell. Again, our building block here is your DNA double helix. So after it wrapped around in your histone proteins, nasan dyan si histone, class, para siyang macaroons here, okay? So here is your histone proteins, and as you can see, parang may bits on a string siya, that's your DNA. So we call that collective DNA plus histone proteins your nucleosome. So one DNA plus histone protein is your nucleosome. So later, we will examine the structure of your nucleosome as well as this linker DNA, okay? So once these nucleosomes pack or tightly packed together, we call it your region ng chromosome, that's your chromatin fiber. So afterwards, ayan, it will be loop or magpo-fold na ang chromatin fiber natin to form your chromosome. Again, class, yung X-like structure ng chromosome, this one is in your cell division, a preparatory step in your cell division. Diba, recall your biology class, okay? So class, also important to remember here, the different types of your chromatin. So iba kasi ang structure ng chromatin natin when it is active or available for transcription and when it is an inactive form, okay? So for our active form, we call it your urochromatin, meaning accessible ang major and minor groove for transcription, replication, and repair mechanisms. So we call naman heterochromatin your inactive form. Clear? Okay, so class, again, we need to dedicate a discussion on your histone proteins. So let's see. Again, your histones, they are types of proteins that is our anchor, okay? The core of our nucleosomes, and of course, ang important or relevance niya in our biological system, particularly in packing your DNA, is to regulate gene expression. So class, here is the structure of your histone protein. Let's see. First one, we have your histone H1 subunit. So this is very important for chromatin compaction and gene regulation. So what is its function? It binds to linker DNA between nucleosomes. So class, somehow, linear yung structure niya. Ayan po si H1 histone. So again, remember the definition of your histone H1 subunit ng histone proteins natin. So this actually appeared in the board exam, including your linker DNA. So class, again, your linker DNA are those DNA segment that we have seen in the previous slides that is between your nucleosome. So parang they resemble a string connecting your beads of your histones, di ba? So for the core ng nucleosome, so this collective structure, DNA plus your histone, is our nucleosome. So class, again, ang basic unit ng chromatin is your nucleosome. And in its core, we have your histone octamer. So when we say octamer, it has 8 subunits po, okay? 2 H2A subunits, so colored in gray here, one in front, one in back, okay? So similar with your H2B, H3, and H4 subunits, they comprise our nucleosome core. So class, each of them has 2 units in this core. Ayan, so again, they are wrapped by DNA. Clear? So class, in our visualization here, ayan ang wrapping natin. This is your DNA double helix. Alright, so class, please remember again your histone proteins. Okay, so maybe some of you confused with our different terminologies. That's why we summarize the relevant terminologies here in our DNA organization. Let's check again. Your gene is a segment of DNA in your chromosomes that can encode a protein or RNA. So sir, simply sa gene are your coding regions. Yes po. So again, we control the expression of traits and your cellular functions. Alright. Second, again sa histone, our nucleosome core, which is a protein around which DNA winds, and of course, it aids the compaction ng DNA and your regulation ng transcription. Thirdly, we have your nucleosome core again class. This is like a bid-like unit of chromatin. So this is DNA plus your histone protein, and of course, we emphasize this, that's the basic unit of your chromatin structure, okay? For the plasmid, sir, parang di na-discuss yan. Class, this is actually an exclusive DNA found in our prokaryotic cells, particularly sa bacteria such as your E. coli. Alright, so this is a small circular DNA in prokaryotes or engineered cells that carries extra genes like your antibacterial resistance or antibiotic resistance, often for survival advantages. So sir, iba pa yan sa chromosomal DNA ng prokaryotic cells? Yes po. Again, your prokaryotic cells chromosomal DNA are the one or is the one carrying the majority of your coding regions. Alright? So second to the last again, chromatin, that's just a region of your nucleosome. So DNA wrapped around histone proteins, which packs your DNA and regulates gene access. And lastly, our DNA organization in eukaryotic cells, that's your chromosome class. So that's just a condensed chromatin structure during cell division. Okay, so class, I know very well familiar na kayo with our central dogma ever since high school. But of course, very basic ang discussion on central dogma during our high school camp or during our senior high school chemistry. But here, as we have seen in our undergrad biochem, there's a lot of things happening in your central dogma, particularly sa replication, transcription, and your translation, diba? There's a dozen of enzymes, a dozen of cofactors, transcription factors, replication factors, and any other factors. It's overwhelming. So don't worry, that's a valid reason. So here naman sa discussion natin, I will emphasize what are the essential information that you need to familiarize, okay? So in the board exam, well, as I have said, majority ng questions are coming from your cellular respiration and your biomolecules. So here, coming from your lipid, all the way to your central dogma, medyo kanaka-naka na lang, di ba? Little concepts about your central dogma, that those are very relevant in your board exam. So don't worry, class, again, we will discuss the specifics of those board exam relevant topics. Again, here in our review, we prioritize your high-yield topics in our crash course discussion. Class, for our central dogma, here is our overview. Ayan. So again, this is a collective process that first converts your genetic information encoded in our DNA into your gene product. So when we say gene products, it can either be RNA or your final product protein in our translation process. Let's see our diagram here. Again, this is your DNA. This will be replicated in our replication, and then it will be transcribed in our transcription process. Check. Of course, class, ang reverse ng transcription is your reverse transcription. So here, instead of starting with DNA, we actually use your mRNA transcript to produce your complementary DNA. So actually, this process is very particularly important on our polymerase chain reaction lab technique. Diba familiar with your RT-PCR? So with that particular technique was used during the COVID-19 pandemic. Going back, from your transcription, we will use your mRNA transcript in order to produce a functional protein in our translation. And class, ayan, so as you can see here, of course, we also have RNA replication. But don't worry, we will not discuss it naman here in our crash course. For the specifics of these three processes, we summarized it here in your table. Check DNA replication, copying of DNA lang po. Okay? So for prokaryotes, you can see here that for all processes, they occur in your cytosol, obviously, because again, they do not have a true nucleus. All right? For transcription, ayan, from DNA, we will now transcribe our genetic info. So particularly, class, this is your genetic transfer process. So here, our product is RNA, and for eukaryotes, both replication and transcription occur in your nucleus. So once done ang transcription, your RNA transcript will go outside the nucleus to your cytosol, particularly in your ribosomes for the translation process. So, diba? Very straightforward from RNA to proteins conversion. Class, ready now with our first process on your DNA replication. Okay, so class, here is our overview of your DNA replication. So again, this is just the copying process of your DNA in order to duplicate our genetic material either before cell division or before replication. I'm sorry, transcription. Class, don't be overwhelmed. Ayan, this is the overview of the processes occurring here in your replication process. And here are the specific info that you need to know. Again, for the location, eukaryotic cell, nucleus po. While sa prokaryotic cell, it's in the cytosol. And here is the feature or are the features for the replication process. So check. When we say semi-conservative, meaning for every rounding replication, our product will contain one new DNA strand and one new strand. Okay? So that's our semi-conservative because again, a part of our strand is an all strand or your parent strand and your new strand is your daughter strand. Alright, for the directionality, sir, same by the 5' to 3' direction? Yes po, class. So that's with respect to your synthesis. So meaning our directionality, di ba recall from our discussion on nucleic acid, it's also 5' to 3' in terms of its structure. Class, again, it's always 5' to 3' direction. And for our last feature, we use your Watson-Crick base pairing to have that high accuracy or high fidelity ng DNA replication. Alright, and of course, ayan, our product or outcome is the formation or production ng two identical DNA molecules. And of course, class, check your manual. How many processes are there for your replication process? Check. Sir, parang 6 ata to. Yes, I think it's 6. But of course, we can divide it into 3 major steps. Okay? So class, that's your initiation. So this one is the assembly of your initiation complex that begins at specific sites. Okay? Next, ayan, your elongation. Basically, we will elongate your existing molecule of DNA through addition of nucleotides. And lastly, ayan, si termination. Of course, our product here is your identical copies of DNA. And here, your initiation complex will be disassembled or magdi-dissociate lahat ng enzymes participating here in your replication process. Or class, there are more than a dozen enzymes actually and other proteins, coenzyme, cofactor that participate in DNA replication. So expect that in our succeeding slides, you will encounter a lot of enzymes and other proteins that participate here in our replication process. So I think you are familiar to some, diba? So because they are very famous, diba? So from our high school camp, we already encountered them. But don't worry, we summarized the specific processes occurring here in our DNA replication. So let's start with your initiation step, the first process of your DNA replication. Ayan, so class again, for your prokaryotic and eukaryotic cells, initiation of DNA replication are different from each other. But of course, ang common theme is that the genome, okay? Our genome, sir, what's the difference of genome and gene? Class, ang gene are the coding regions only, okay? Ang genome is our complete set of DNA. So meaning it includes both your genes and your non-coding regions, okay? So in short, a genome contains all the genes, while your gene is just a specific segment of your DNA that can code for a protein or RNA. Alright, so going back again, our genome is actually divided into replicons. So class, si replicons ang site ng beginning ng initiation. Again, it has an RE site kasi or your origin of replication. So si RE or RE site or origin of replication, they are all just the same thing, okay? And of course, we also have a termination site. Let's compare our initiation process for prokaryotes and eukaryotes. Firstly, sa prokaryotic cell, your RE or RE site is only a single defined origin. Comparing it to your eukaryotic cells, ayan, of course, because very large or extended ang chromosome natin, we need multiple RE sites, okay? So that's why in terms of the replicons, kung one lang ang RE site that is in your replicon, meaning one replicon lang din po per genome of your prokaryotes. So for eukaryotic cell, because we have multiple origins, sir, meaning meron din tayong multiple replicons, yes. And that actually gives us or gives your replication the efficiency in terms of its speed. O diba, let's compare. Because only one RE site and one replicon for your prokaryotic cell, of course, the speed of replication is generally faster. Well, comparing it to your eukaryotic cell, well, slower per origin siya, but of course, because it is simultaneously occurring per replicon, yan, overall efficient naman siya due to multiple origins. Alright, so class, here is an image, okay? An electron microscope image of your different replicons for prokaryotic and eukaryotic cells. So let's check. First, for your prokaryotic cell, ayan, very obvious ang ating replicons here. So actually, this is during the replication process now. That's why the elongation of your daughter DNA is currently ongoing. Okay, so again, class, one replicon meaning one RE site. Okay, so class, we call this actually your replication bubble. For our eukaryotic cell, of course, here you can see linear chromosome, and thus very efficient in replication because it has multiple origins. So let's see. This one is your RE site. So actually, this one is a replicon. This one is also a replicon, and this tiny region is also a replicon. Okay, so very obvious ang multiple origins and multiple replicons for our eukaryotic cells. Okay, so class, let's now discuss the specifics of your initiation process, starting with the assembly of our initiation complex. All right, so class, again here on our first step, we will discuss the components of our initiation complex already. Ayan, so first, of course, as we have discussed earlier, we have your RE site. Di ba recall, for your prokaryotes, this is an only one site. For your eukaryotic cells, so these are multiple, right? So this is specific site where DNA replication begins. So class, yung sequence ng RE site in terms of its DNA sequence is usually rich in 80 base pairs. Also, what's the reason? Di ba recall, your 80 base pairs compared with your GC base pairs, they only have 2 hydrogen bonds. So GC, it has 3 hydrogen bonds. So relatively much easier to unwind ang ating 80-rich base pairs. Clear? So of course, that's with the help of your initiator protein. So that's your DNA. So yan talaga yung name. So class, actually this stands for your DNA replication initiator A protein. Okay, so again, this binds to the origin of replication and it initiates the unwinding of DNA to start replication via yan. Magre-recruit siya ng replication machinery. Class, yung replication machinery here are your enzymes that will help in unwinding and in binding our specific catalysts or enzymes here. Okay, so as you can see here class, ayan ang ating replication machinery or our initiation complex assembly. Diba? So later on, we will discuss these two important component or enzymes or proteins rather. So class, before we discuss these two, of course, let's also discuss the characteristic structure of your initiation complex. Diba? So here, we have your replication bubble, which is the localized unwound region formed at your origin. So here you can see this is a bubble. Ayan, simply a bubble. And of course, at the ends of your replication bubble, we have Y-shaped structures where replication proceeds bidirectionally. Okay? So class, again, complementary kasi ang strands. That's why our replication will proceed 5' to 3' from both strands or to both strands. Okay? So nasan dyan yung Y-shape? Parang hindi naman clear. So this is the first Y shape. Ayan. So of course, it looks like letter Y. So for the second one, ayan. So this is our replication fork. Okay. So class, again, these are the two important proteins and enzymes that will help in completing our initiation complex. The first one is a helicase and it's fully responsible in unwinding your DNA helix. So bidirectional din siya. One is going here and one is going here towards this direction. Okay. So class, important that your helix or your DNA helix is exposed, particularly your template strands, these two template strands to our enzymes to start the elongation process. And of course, to aid the activity of helicase, we have your single-stranded binding protein. So literally from its name, it stabilizes your single-stranded DNA. O sir, paano po? So class, of course, this region, your replication bubble, has the tendency to reanneal or to reattach again to their specific base pair. So SBPs will prevent it from happening. Okay? So class, this is our, again, the initiation complex. Before we proceed with our next step, let's just recall the difference between your replication bubble versus replication fork. Because I think hindi clear yung difference niya from our last slide or the previous slide. So again, class, this is our bubble. Okay, so these entities here are just your initiation complex. So helicase, SBPs, DNA initiator protein. All right, so class, here is the bidirectional replication process. So one is going to your 5' to 3', and another one will also go to your 5' to 3' direction. Sir, in terms of synthesis, no? Yes po. That's your replication fork. Okay, so class, please remember the difference between your replication bubble and replication fork. Okay, so that's it. We completed our initiation complex assembly. So now our regions or our RE sites are now ready for your primer synthesis. So class, from the name itself, your primer will prime the start of elongation. But technically, class, this is still a part of your initiation. It's like a preparatory step for your elongation. Now let's check. Again, the enzyme that is responsible to your addition of primer is our primase, particularly your RNA primase enzyme. So literally, it will synthesize short RNA primers that will serve as a signal actually to your DNA polymerase or to initiate DNA elongation process. O class, locate your RNA primer here for both strands. It always occurs from the 5' to 3' direction. Or see class, again, 5' to 3'. That's why our synthesis is bidirectional. Okay, so again, your primers provide a free 3' hydroxyl group for DNA polymerase to elongate or to extend the nucleotide chain. So actually, class, that is also the reason why our directionality in terms of the synthesis is from 5' to 3'. Sorry. So recall, in our 5' end, we have a free phosphate group, and in our 3' end, we have your free hydroxyl group. So your free hydroxyl group is actually reactive. Okay, so it will initiate an attack to your phosphate group in your existing nucleotide chain, and thus it serves as your chain elongation reaction mechanism. Okay, so as again, in our preparatory step or the primer synthesis in our initiation, we also need your polymerase. So part siya ng initiation, but it's also a large part, diba? A large part on the process of elongation. So your polymerase enzyme is responsible in elongating or extending your RNA primers with DNA nucleotides. So again, from 5' pa rin to 3'. So si primer, hindi na yan papalitan. Class, wag ma-excite. Later, we will also discuss the primer removal to be replaced by our original nucleotides from your DNA polymerase. And lastly, class, remember, this is actually a board exam question. So DNA topoisomerase or class, don't forget, it's alias, diba? Your gyrase enzyme. So your topoisomerase or gyrase, ayan, it relieves the supercoiling. So it's ahead of our replication fork, diba? Of course, kailangan niyong i-relax yung DNA chain ahead of your helicase for unwinding. Okay? Kasi class, ang unwinding, it induces supercoiling at the upstream. Okay? At the upstream side of our helicase activity. Okay? So I hope that's clear. Again, This is your topoisomerase. Please remember your topoisomerase. All right. Ayan. Now, we are on our second main step, the elongation step. Check your manual. Sir, we actually have two key steps here. Yes, because again, we have two different nature of your strands. One is your leading strand and one is your lagging strand. Familiar, class? Yes, sir. Okay, again, class. Sa leading strand, much easier po ang DNA elongation because here, it's just a continuous synthesis in the direction of your replication fork. And of course, if this is continuous and in the direction of the replication fork, in the lagging strand, ayan, you can see it is discontinuous and opposite to your fork movement as Okazaki fragments. So class, your Okazaki fragments are also added by your DNA polymerase via the 5' to 3' direction. So this is the direction of movement of your replication fork, and for the lagging strand, ayan, it's opposite your movement ng replication fork. So here, let's first discuss the leading strand synthesis. So class, remember, very specific, ang isocyme ng polymerase that catalyzes our DNA elongation. So here we have your DNA pol tree, a very busy enzyme. You can see, again, always 5' to 3' ang direction ng addition ng ating deoxyribonucleotides. Okay? So again, class, ang hint here for leading strand, it's always your continuous synthesis. Okay? So again, class, ang directionality niyan is always following your helicase enzyme. Okay? So it synthesizes your deoxyribonucleotides toward the replication fork. Okay, so class, let's proceed with, ayan, amine. So this is just a parallelism of what's happening in your leading strand. Again, helicase, nasa unahan niya ng chain in order to initially unwind your DNA helix for the activity of your DNA polymerase tree in your leading strand. So again, class, continuous po ang synthesis in your leading strand. Sir, do we need to memorize the subunits of DNA pol tree? No po. Okay, so ang important here is you know the difference between the functions of our DNA polymerase. But don't worry, we will also discuss this here in our replication process. Okay, so class, let's continue our discussion on your lagging strand synthesis. Again, class, discontinuous po siya because here, our template strand is actually your 5' to 3' template strand. Okay? That's why, of course, very hard for our DNA pol to add your Okazaki fragment. So actually, it's a fragment by fragment ang addition here. That's why we call it your discontinuous DNA synthesis. So class, still the same ang mechanism ng primaries natin. For both leading and lagging strand, they always initiate the addition of your primers. So again, this primers kasi act as your signals for the activity of your DNA polymerase 3. Again, class, for both the leading and the lagging strand. So class, here as well in your lagging strand, the DNA pol tree is the one responsible for elongation via the synthesis of short DNA segments. So class, here is your Okazaki fragments. Again, we add your Okazaki fragments little by little. O sir, anong mangyayari? May gap yan. Class, later sa ating termination step, we will also discuss how to seal the gap between your phosphodiester linkages here. Alright? So lastly, we have your sliding clamp. Kung si helicase, meron siyang katandem, si SSBPs, of course, sa DNA polymerase tree, in order to continue its activity, it needs your sliding clamp to hold its structure in place during the fragment synthesis. Okay? So again, class, recall, here your sliding clamp for DNA polymerase tree. Alright, so class, before we discuss the next step, this is the specific reaction that occurs upon addition of your deoxyribonucleotides, regardless if it's in your leading strand or lagging strand. Let's check. Again, the direction of synthesis from 5' to 3'. That's why here you can see the initiation of attack of our free hydroxyl group at the 3' position. Check. Si lone pair ng electron in your oxygen in the hydroxyl group will initiate an attack to your incoming deoxynucleoside 5' triphosphate. And of course, ayan, we will liberate your pyrophosphate in order to successfully add your incoming DN-TPs. Okay? So class, again, for both lagging and leading strand, your mechanism is just the same. Clear? Ayan, before we proceed, I would like to highlight lang your sliding clamp here. Again, the sliding clamp is very particularly important for the continuous activity of your DNA polymerase tree. But class, of course, for your sliding clamp to hold your DNA polymerase in place in order to load it, diba? Meron tayong clamp loader. So si clamp loader will attach your sliding clamp to our DNA replication site. Okay? And there, as you can see, upon attachment, your DNA polymerase will bind to it in order to stabilize its structure. Clear? Again, class, no need to memorize the clamp loader, but please remember, with your helicase, we have SSBPs. Here in your DNA polymerase, we have your tandem molecule, your sliding clamp. Alright, so class, this is the step that I am pertaining to earlier, diba? So of course, upon addition of primer, we actually need to remove it, okay? Hindi siya ma-retain sa structure ng ating elongating strands. So here, the polymerase responsible for the removal of primer is your polymerase 1. So this is through its 5' to 3' exonuclease activity, meaning it will cleave your phosphodiester linkages from your sugar phosphate backbone. Again, your 5' to 3' exonuclease activity is specific for your polymerase 1 enzyme. And after remove, ayan, siya rin po ang magtatrabaho to replace them with your original DNA, DNTPs via its 5' to 3' polymerase activity. So sir, meaning two catalytic sites ang meron sa polymerase 1. Actually class, for all polymerases, they have numerous catalytic site. So that's very important in order to prevent errors or what we call mutations in your DNA replication. So very notorious kasi ang mutations here in your replication. O see, here is the addition of your original DNA upon removal ng DNA primers. Again, from 5' to 3' direction. O sir, anong mangyayari sa mga gaps in your sugar phosphate backbone? Again class, ang gaps here is in your sugar phosphate backbone. Don't worry. Ayan, ilaligate yan ni DNA ligase in our 6-step or last step ng elongation process. O see, this will sail or the next or the gaps between Okazaki fragments through your phosphodiester bond formation between the 5' phosphate and 3' hydroxyl ends. Clear? Again, class, this ligase activity is specific for your lagging strand because that is where the Okazaki fragments were added, diba, sa elongation step. Alright, so class, again, we have different isozymes of your DNA polymerases and each of them has their own very unique and different functions. So they catalyze the elongation of DNA strands after replication. O sir, always silang nagpa-participate sa elongation, no? Yes po. And again, for majority ng bacteria, such as your E. coli bacteria, they have 3 main types, okay? So each of them, again, has specialized roles in replication and repair. So let's check. Ayan, from DNA pol 1 and DNA pol 3, we discussed this too. But remember, we also have your DNA pol 2, mainly for repair. Let's check. Class, yung polymerase activity is responsible for the elongation process. So with this activity, all of your polymerase isozymes are capable of your 5' to 3' polymerase activity because of their main job description, no, diba? Their main job responsibility is in your elongation process. That's why it's already a given, okay? So for our next activity, your 5' to 3' exonuclease activity, so class, synonymous yan sa removal ng primer. And recall, the one responsible in your primer removal is only your DNA pol 1, okay? That's why this is only the polymerase isozyme capable of your primer removal activity. That's actually its main job description, okay? And then lastly, we have your 3' to 5' naman, exonuclease pa rin for proofreading. O sir, lahat sila capable? Yes, class. That's a very important activity or function for all polymerases in order to assure the high fidelity of your replication process. So class, remember, ang proofreading always from 3' to 5'. Sir, bakit po? Because ang inyong polymerase enzyme will not slide back to the 5' position. Instead, it will have an activity or they will have an activity via their 3' to 5' exonuclease activity for efficient proofreading mechanism. Okay, so to summarize again, DNA pol 1, main job description, primer removal. Pol 2, DNA repair, and lastly, si pol 3, mainly for your elongation step, as we have seen in our discussion earlier. Okay, so class, to summarize our elongation step here, ayan, and again, for your leading strand, it's a continuous synthesis. For the lagging strand, this continuous synthesis via the 5' to 3' addition of your Okazaki fragments. And remember, we have different isozymes that can extend your primers, can remove your primers, and can repair your existing DNA chain. And also, class, don't forget, your RNA primers will be removed and replaced with DNA, okay? So major confusion kasi yan ng students that they thought your RNA primers will be included in the final product. Again, we have a primer removal step in our elongation process. And to end the elongation step, ayan, isisil na ni ligase ang phosphodiester gaps in your lagging strand, okay? Very specific ang activity ni DNA ligase in your lagging strand. And class, ayan, here's the summary of our elongation complex assembly. As you can see, this is a very busy process. So again, simultaneous po to each replicons na go-occur ang ating elongation step, okay? So that's why kasi, or this is the reason, bakit maraming replicons ang eukaryotic cells for efficient replication. Of course, kapag isa lang yung replication, puti na ang mata ng helicase enzyme natin, hindi pa siya tapos, di ba? So class, to end our discussion on replication, ayan, may pahabol tayong step. The termination process where this occurs when your replication forks finally meet its specific termination site. So class, ang ginagamit niyang hint is your TUS proteins or your termination utilization sequence. So class, this specific protein kasi will block or inhibit the helicase activity. So pag na-inhibit sa helicase, down ang unwinding, okay? It will stop unwinding your DNA helix, and thus the replication machinery will dissociate and will release it upon binding to your TUS proteins, okay? So class, again, this is a specific termination site where your TUS will bind, and of course, ayan, helicase activity will be blocked. Thus, they will dissociate along with their single-stranded binding proteins. This will halt the whole replication process. For the termination, all you need to know, ayan, the termination sites are the specific sequences where replication ends and your TUS protein binds to this termination sites and finally blocks your helicase activity. All right. So class, again, as we have discussed earlier, very accurate ang DNA replication process, both in prokaryotic and eukaryotic cells. If there are mutations, of course, those are involved in the evolution of your organism. So those are the origins of our evolution. Okay, so class, here is our Did You Know segment for the DNA replication. And this is related to your high fidelity or high accuracy ng replication process. Or did you know that DNA actually makes fewer than 1 mistake per billion letters or per billion nucleotides copied in our DNA replication? O, diba? Amazing. Oh my God, wow. That's like perfectly transcribing 100 full dictionaries with only one typo, diba? Unbelievable. So here, of course, class, take note, errors are very, very, very low, but it's never zero. Okay? So this is the reason bakit meron tayong low final error rate. This is a collective external mechanism for your accuracy or high fidelity ng DNA replication. First one, our first line of defense, ang base pairing via your Watson-Crick base pairing and your Chargaff's rule. Second line, you have the activity or the proofreading activity of your 3 different polymerases during the replication process. So ewan ko na lang, di ba, kung may makalusot pang mutation. And lastly, kapag merong nakalusot, we have your different repair mechanisms. So, di ba? This is a third layer levels of your DNA accuracy or DNA replication accuracy. And that's why we will also discuss the fidelity of your DNA replication. Again, class, for the fidelity, this actually has an evolutionary impact, okay? So, of course, this prevents large or impactful mutations, but some mutations also persist. And these are sources of genetic variation through natural selection. But here, class, again, we will just focus on the high fidelity or accuracy through your different mechanisms. First one, again, is through specific base pairing. Here, what's syncric base pairing? Second is the proofreading activities of your different polymerases participating in our replication process. And of course, if there's still errors upon termination of replication, of course, we still have your repair mechanisms in order to ensure genomic stability. So again, class, those contribute to our low final error rate. So remember the three again. Your specific base pairing will ensure correct nucleotide pairing. Or remember the discussion on our nucleic acids and their key properties, diba? So one of the key properties in your DNA is the specific base pairing. Sorry. So second one, we have the proofreading activity again via the 3' to 5' exonuclease activity of your polymerases to remove the mismatches. O diba kanina I emphasized that this 3' to 5' or the reverse direction is very important in order for your polymerase to not slide back all the way to your 5' to 3' direction. Okay? And lastly, we have your repair mechanisms to fix your replication errors. And sir, ano pong i-focus natin? Class, we will now focus on our different repair mechanisms depending on the replication errors. Again, class, in your DNA, your fidelity, ayan, it just refers to the accuracy with which our DNA is copied during the replication. So this is very important to ensure the correct sequence and to maintain our replication with minimal errors or mutations. So later on, we will discuss the types of mutations that can occur in your replication process. Again, this will be a domino effect kasi. That's why it's very detrimental. Okay? So class, to discuss our different DNA repair mechanisms, so here are our different errors upon replication. So here, your mechanisms will correct replication errors and DNA damage, again, mainly to preserve the integrity of our genome, our whole DNA information, okay? And of course, secondly, to prevent mutations, because again, mutations are domino effects. Starting from replication, it will be carried until your translation. Now check. The first error is actually your single strand break or your single base damage, meaning in a certain strand, your single base was damaged or it broke. Okay? So here, our repair mechanism is through your base excision repair. Second, ayan, formation ng bulky lesions or your crosslinks. Class, example of our crosslinks involve the creation of your pyrimidine dimers. Also, what's happening here? Class, your pyrimidine in particular can cross links to each other through formation of your dimers. Okay? So those are induced via external factors, or we call it your mutagens. Okay? And here, our biological system combat it through your nucleotide excision repair. So meaning a large portion or region in your newly synthesized DNA will be cut or excised in order to remove that particular dimerization or pyrimidine dimer. Okay? And lastly, second to the last, we have your base mismatch. Okay? So here, class, remember, a base mismatch, that's just simply or literally mali ang base pairing natin. Okay? So here we have your mismatch repair. Class, please do note that these three are the most important. So double strand break, ayan, supplementary discussion na lang yan, where both strands has been damaged or have been damaged. So parang ano lang siya, similar with your single base damage, but here, ayan, for both your new strand and old strand, there has been a damage. And of course, we have two different mechanisms, via homologous recombination or through non-homologous and joining mechanism. So class, don't worry, these two naman are not that important. That's why we will just focus on these three different mechanisms, particularly kay nucleotide excision repair and mismatch repair. Let's see the summary of your different mechanisms. Again, for base excision, this repairs your small non-helix distorting base lesions. As you can see, very particular lang yan sa single base repair. Okay? So nucleotide excision, again, ang clue natin dito, class, is the removal of your large portion ng newly synthesized DNA double helix. Okay? So that's to remove, okay? That is to remove your bulky DNA lesions or your thymine dimers in particular. For mismatch, of course, from the name of your mechanism itself is to correct the base pair mismatches after your replication. So sir, meaning yung base pair ang nagka-error dito, no? Yes, class. Okay? So of course, spontaneous naman na magkaka-error during your replication. Hindi naman perfect, di ba? Your replication machinery is not perfect. They are still prone to commit errors. And lastly, for your double strand break, again, this fixes breaks via homologous recombination or non-homologous end joining. So what does that mean? So meaning, class, we will just rejoin the two ends where your double strands were damaged. Okay? So class, for the sake of our discussion, again, we will just cover the specific mechanisms of the three different repair mechanisms. Your base excision repair, your mismatch repair, and your nucleotide excision repair. Starting with our base excision repair. Again, ang target natin here is to remove that small base lesions by excising the base first, then the backbone. Sir, anong meaning nun? O class, ang meaning nun, we will first excise your nitrogenous base via the damage or the degradation of your hydrogen bonds. Okay? So that's through your glycosylase activity. And here we will create an AP site. So this is also called your a basic site, which is a location in your DNA where a base is missing. So here, class, ayan, the sugar and phosphate backbone is still intact. That's why we need your endonuclease. Class, the endonuclease or any nuclease, their target point of degradation is to disrupt your phosphodiester linkage. That's why after the glycosylase activity, your endonuclease activity will degrade the phosphodiester linkage or it will disrupt it. And here, ayan, ito na yung point of mechanism or repair natin via short patch path or your long patch path. So class, ang difference lang here is that kapag single base lang ang affected, that's the short patch. So here we have different enzymes, your polymerase for the addition of nucleotide, your endonuclease for the creation of your phosphodiester linkage in one end, and your ligase to seal the gap to complete our base excision repair mechanism. So the same mechanism can take effect in your long patch, but here the DNA polymerase enzyme will add series of nucleotides before the endonuclease and DNA ligase perform phosphodiester bond formation. Clear? Again, class, for endonuclease and DNA ligase, their point or their target mechanism is through the creation of phosphodiester bonds. Are clear? Ayan, for our second repair mechanism, again, this is for your large portions ng repair natin or your damage. Particularly, very important, sa bulky DNA damage or your creation ng pyrimidine dimer. So here, class, we will cut a short surrounding strand for your DNA double-stranded or double helix. So see, this is a typical pyrimidine dimer or thymine dimers. It creates a bulky structure here or your bulky lesion. And of course, ayan, literally, ikakat natin yung certain region in order to open up your DNA strand to form a new bubble. And here, ayan, we already excised your DNA or your damaged DNA. And of course, again, the polymerase will extend it, and then the ligase will ligate your strands via its phosphodiester bond formation. Okay, and lastly, we have your mismatch repair. Sir, unique yan? Yes, class, because its mechanism is far different from the first two repair mechanisms. So let's formally define mismatch repair. So again, this corrects base pair mismatches after replication by identifying first the newly synthesized strand, and then afterwards, it will replace the incorrect bases with the correct ones. So actually, class, same activity. The exonuclease activity will be used here in order to repair your mismatch ng base pair. Okay, so let's check the specific mechanism. So class, again, this is very particular for your DNA Pol 1. So here, you have your 3' to 5' exonuclease activity. So this is the active site colored in pink. And the polymerase activity, ayan, colored in blue. Again, ang polymerase activity, 5' to 3'. Ang exonuclease, in order to prevent from sliding back, your DNA polymerase will use its 3' to 5' exonuclease activity. Let's check. First, upon addition now ng DNA polymerase, your tautomeric cytosine pairs with adenine. Class, again, your tautomer or tautomerization is the formation of your keto-enol functional group. And this is capable for cytosine to pair with adenine. And here is the mismatch or the mispaired nucleotide with your cytosine pairing with adenine. And of course, ayan, immediately, madedetect yan ni DNA polymerase. That's why it will slide back to the position in the mispaired base in your exonuclease active site. Again, the exonuclease active site will be the one to perform the proofreading activity to remove our mispaired nucleotide. And again, ayan, i-elongate na ni DNA Pol with the correct base pair here in order to continue your replication process. All clear? Ayan, so class, we now end our discussion on DNA replication by this summary slide. Again, this occurs through 3 steps: initiation, elongation, and termination. Class, please remember the key features we discussed, ha? Again, sa initiation, ang main clue natin dyan is the formation of your initiation complex via the activity or the origin of replication. Okay, so again, this begins at the RE site where your helicase will bind to unwind your DNA, and then your SSBPs acts as your support protein to stabilize your strands and to support the activity of your helicase. So, anong product? Of course, that's your unwound or open DNA. And then, from your elongation process, we will have your main character enzyme here, the DNA polymerase, will elongate our existing strand. And class, in particular, always remember, leading strand, continuous synthesis, lagging strand, discontinuous synthesis via the step-by-step or step-by-the-step, diba? Addition of your Okazaki fragments. And here, our temporary intermediate is your 2 elongating daughter strands. And lastly, for termination, we have your dissociation ng ating replication machinery when your termination sites have been reached by your TUS protein or your termination utilization sequence. As a result, the helicase activity will be inhibited by your TUS upon binding in your termination site, and the product, ayan, our final product is 2 identical double-stranded DNA molecule. Again, class, from your parent molecule in the initiation step, we will unwound this in order to separate the strands for the elongation step, and finally, upon termination, we produce 2 identical daughter double-stranded DNA molecule. And here, semi-conservative siya again because each consists of 1 parental strand and 1 new strand. All clear? Okay, so class, that's it for our DNA replication. So to end our discussion here, I prepared a set of quick questions. Diba? So are you familiar with the game Emoji? Diba? I will give you a certain group of emojis, and then you will guess what does this emoji depict. First, this is our topic earlier, diba? So this is your DNA replication. Class, again, copying your DNA occurs in your DNA replication. Next item, sir, ano yan? Class, check. We have your chrome, we have your matte, and we have your tin can. Sir, chromatin can? No, class, chromatin lang po. Okay? So this is the basic building block of our chromosome, our DNA organization for your eukaryotic cell. And lastly, ayan. Sir, snake ba yan? Yes. And rock? Yes as well. But of course, class, that's not our missing term here. So class, the sound of our snake, hiss, diba? And our figure here, that's actually your stone, diba? So this is actually your histone protein. Okay? So that's our mini icebreaker. Okay? So class, again, we will now start with our next topic. After replication, we have your transcription. So you can pause this video if you need a 5-minute break in order to have your sanity break, diba? So of course, class, you can have your short breaks or CR breaks, hydration breaks. So I know, nasa last stretch na tayo ng discussion. That's why, very super saturating na ang information on biochem. O diba? Loaded with information, class. But here, again, very minimal ang calculation. That's why, very hit or miss in the board exam ang type ng questions in your biochem. Here in our crash course, we are targeting or we are focused on giving you your high-yield topics that are sure to appear in your board exam. So class, at this point, I will end the video first, okay? So I will cut our discussion on Crash Course 9 into 2 videos. So this part 1, we already finished discussing your overview, particularly the DNA organization and your DNA replication. So on part 2, we will continue our discussion on transcription, translation, and mutation. So see you on the next video, class. Bye!